Hello friends, welcome to my channel. Today we are going to discuss some of the terminologies used in plant tissue culture. So let's see the first, that is totipotency. So totipotency, it is one of the important term which is used in plant tissue culture. Without that ability, the science of plant tissue culture, it is impossible. So totipotency means it is the ability of plant cell to grow into the whole plant. For example, if we take the cells like collenchyma, parenchyma, anther, embryo, endosperm, so that cell have the ability to grow into the whole plant body. So that ability which is present inside the plant cell is called as an totipotency. The next term, that is the explant. So what is explant? So explant, it is the any plant part which is used to propagate the whole plant body in the plant tissue culture. So that plant part in the tissue culture is called as an explant. Then the next term, that is the callus. So callus, it is an unorganized mass of cells. Means when we put up the explant on the media for its further development, after some weeks the media shows presence of the mass of cells. The mass of cell, it is without stem, without leaves, without roots. It is only the collection of cells. So that mass of cell in tissue culture is called as an callus. Then the next term, that is the media. Every plant requires nutrition for its own growth and development. So here in the tissue culture, we provide the artificial nutrition in the form of media, and basically the MS media, that we call it as an Morashinge and Spook media, it is used for the growth of the plants under the tissue culture. Then next one, that is the suspension culture. So suspension culture means it is the type of liquid culture. Basically, the agar is used as a solidifying agent to solidify the media. But when we skip the use of agar in the media, so that type of media or that type of culture is called as a suspension culture. The next one, that is the anther culture. When we use anther as an explant to propagate the plants into the whole plant body under the tissue culture, so that type of culturing is called as an anther culturing. This type of anther culturing, it is also called as an haploid cultures. The next one, that is the embryo culture. When we use embryo as an explant for the growth of the plant body, then that type of culturing is called as an embryo culture. This is also one type of haploid culturing because it shows presence of haploid chromosomes. The next one, that is the ovary culture. So ovary culture means once again when we use ovary as an explant for the growth of the plant body, then that type of culturing is called as an ovary culture. The next one, that is the endosperm culture. When we use endosperm as an explant for the plant propagation under the tissue culture, so that type of culturing is called as an endosperm culturing. Then the next term, that is the hardening. Hardening means when we remove the tissue cultured plants from the aseptic condition and locate them into the greenhouse or polyhouse. So removal of that tissue culture plants, that process is called as an hardening. So these are the some of terms which are related to the plant tissue culture. If you like that video, please like, share and subscribe my channel. Thank you.